Good afternoon, everyone. As Provost of New York University, it is my privilege to welcome you to this afternoon's lecture. I am pleased that you have joined us for this wonderful opportunity to hear a sample of the work of Dr. Ada Yonath. I would first like to take a moment to acknowledge Mrs. Helen Kimmel, who is with us here today. As a trustee of New York University, Helen's leadership and generosity to NYU and its NYU Langone Medical Center, in partnership with that of her late husband Martin, are paving the way for breakthrough research by our most brilliant scientists, supporting individual scientists and their research projects, both here at Washington Square and at the Medical Center. In addition to her leadership positions at NYU, Helen is also a life member of the Board of Governors for the Weizmann Institute of Science, where our speaker, Dr. Yonath, occupies the chair that bears the names of Mr. and Mrs. Kimmel. So thank you, Helen, for all you have done to advance science at NYU and around the world. In a few moments, you will hear from Professor Tamar Schlick about Dr. Yonath's scholarship and her talk today. But before that, I would like to briefly introduce Professor Schlick to you. Tamar Schlick is a professor of chemistry, mathematics, and computer science at New York University. She received her PhD in applied mathematics from the Courant Institute of Mathematical Sciences, and after a postdoc fellowship at the Weizmann Institute, working with a grand pioneer of molecular mechanics, the late Shanir Lifson, she joined the faculty of NYU. Professor Schlick is recognized for developing innovative mathematical and computational tools for biomedical modeling and simulation, including a long time step integrator, a multivariate minimization algorithm, and coarse-grained models, and applying them to generate atomic-level insights into fundamental molecular processes, including the structure of DNA and RNA. She has 150 published papers, is a member of several editorial boards and advisory committees in mathematics, computational biology, and chemistry, and is active in the community by organizing many workshops, conferences, and new programs, especially to enhance and broaden graduate education. She has trained about 35 postdoctoral fellows and 35 research students, as well as a dozen undergraduate and high school students. Among Professor Schlick's honors are a Guggenheim Fellowship, a Sloan Fellowship, a Howard Hughes Medical Institute Investigatorship, and an NSF Presidential Young Investigator. Her textbook, Molecular Modeling, an Interdisciplinary Guide, is widely used in modeling computational chemistry and biophysics courses worldwide. The second edition of that textbook is due out later this spring. Professor Schlick is also a founding member of WOW, Women on Women, a supportive group for women in the sciences here at NYU, which along with the Department of Chemistry and the Dean of Science is a sponsor of today's event. So thank you, WOW, the Chair of Chemistry, Michael Ward, and the Dean of Science, Dan Stein, for this afternoon's event. And now, please join me in welcoming Professor Tamar Schlick, who will introduce today's speaker. Tamar.
Thank you. Okay, thank you very much and for the introduction. It is really a great pleasure for me to introduce to you my friend and colleague Ada, who is an extraordinary woman and scientist, who also holds a special relationship with the city and university. I can use this. Ada has visited she also attended a prior event sponsored by WOW and the Dean of Libraries to celebrate James Watson's 80th birthday. Today, she will share with us, yet again, her deep insights and expertise concerning the ribosome structure and function. As way of background, Ada was born in Mea Shearim in Jerusalem. At age 13, after the death of her father, she moved to Tel Aviv with her mother and little sister Nurit, today a successful psychologist. To help her mother, Ada worked in many jobs, from cleaning stairwells to giving private lessons. She was accepted into a prestigious high school, and in order to pay the high tuition, she tutored new immigrants. However, Ada had to give up her studies when she found a higher-paying job in order to support her family. Realizing how special Ada was, the school headmaster nonetheless managed to bring Ada back to school when she found her a job at the school. After her obligatory military service, Ada gave up her wish to live in a kibbutz in favor of chemistry. Ada received her bachelor's degree in chemistry and master's degree in biochemistry from Hebrew University, Jerusalem, and PhD in X-ray crystallography from the Weizmann Institute in Rehovot, Israel. After postdoctoral positions at the Mellon Institute in Pittsburgh and MIT, she joined the faculty of the Department of Structural Biology at the Weizmann Institute, the crème de la crème of Israeli universities, and initiated protein crystallography there. Significantly, this was the only such laboratory in the country for almost a decade. I just returned from a visit there, where I enjoyed talking to many scientists from across the world, as well as seeing and smelling many gorgeous spring flowers that surround the campus, as you can see from these beautiful photos. The beautiful flora is equally matched by the special people who work there. Ada continued to work very hard at her science because she chose a very challenging problem, deciphering the structure and function of the ribosome, the RNA protein complex that is the cell's workhorse, making proteins. After obtaining preliminary promising crystallizations, it took Ada 8 years to develop a systematic protocol to obtain ribosome crystals suitable for crystallization analysis. That was a feat on its own right, which revolutionized structural biology. 12 more years followed until Ada revealed the ribosome structure in full glory in 2000. Throughout this time, Ada had to seek funding to support her group's laborious work, and later on, 15 years after she started, she also had to face other competitors. Some rivals criticized her approach and reduced the value of her efforts. Unfortunately, that is the sign of a brilliant scientist. But Ada persevered. Ada says of those years, during those difficult years, I learned a lot about humankind. I thought I'd get to age 65, retire, write a romance novel on the psychology behind all the science, gossip. But as of yet, I have not retired. I should add that Ada has not even slowed down. At Weizmann, Ada quickly rose through the ranks, becoming department chair and later director of the Kimmel Center for Biomolecular Assemblies. Mrs. Kimmel, whose name adorns this building at NYU and who is here with us today, as already mentioned by our provost, has been most generous not only with Ada's center at Weizmann, but the square here and with the NYU School of Medicine. At the same period, Ada also directed a Max Planck research unit in Hamburg, Germany for 18 years until 2004. For her heroic doggedness in solving the structure of the ribosome and interpreting its function, Ada has been recognized by numerous honors and prizes. These include the Israel Prize, Rothschild Prize for Life Science, Wolf Prize, Louisa Gross Horwitz Prize from Columbia University, Paul Ehrlich Ludwig Medal, Linus Pauling Gold Medal, UNESCO L'Oreal Award for Women in the Life Sciences, Albert Einstein Award, World Award of Science, Eris Prize for Peace, and of course, the Nobel Prize Chemistry in 2009 with Venkatrama Ramakrishna and Thomas Stites. Ada has also received an honorary doctorate from Oxford University and is a member of the U.S. National Academy of Science, European Academy of Sciences and Art, and the Israeli Academy of Science and Humanities. Many of us in the scientific community have known and recognized Ada's work in structural biology for a long time, but the Nobel introduced her to many people across the world. As an Israeli, I have enjoyed following an entire country's growing fascination and delight with Ada, the first Israeli woman to win the Nobel and the first woman to be recognized by a Nobel Prize in Chemistry in 40 years. You can get a glimpse of this from these headlines that decorated daily newspapers and magazines of all kinds in Israel. To honor Ada, we have with us today Hilla Elroy, the advisor to the Council General of Israel, who came with Asaf Shamis and Muriel Schottlander, experts in politics and internet technology. Flattered by all these prizes, Ada also admits that while rewarding, the real prize is the excitement seen by scientists, especially young researchers, as they make discoveries. Ada adds that young scientists need daily encouragement, especially when results are not immediate. Scientists, she says, need peace to work on their projects and a lot of patience and persistence. Just before the Nobel ceremonies, Ada said, I heard that there was a survey in high schools throughout Israel, and many now wanted to be chemists. This is the best thing that came out of this Nobel Prize. Ada's close friend and renowned crystallographer, Tzipi Shaked, describes Ada as a scientist and a mensch. The latter is an untranslatable Yiddish word that does not, unfortunately, describe many scientists in competitive research areas. Harold Chiraga, emeritus professor of chemistry at Cornell University and fellow mensch, has also known Ada for a long time. Recently, he told me, Ada used to stay with Miriam and me when visiting Cornell to use the Cornell synchrotron. But staying with us meant only that Ada's bag stayed in the guest bedroom. Ada, of course, spent her nights at the synchrotron. I hope I have given you a glimpse of how extraordinary Ada is as a person and a scientist. Before giving her the floor to talk about her discoveries, I'll end with a movie that the WOW Steering Committee and I have prepared on Ada's life. Thank you, Meredith, Namhi and Rosanna for putting this together, and also Coldplay for writing this beautiful song called The Scientist. I also want to thank Julie Kaplan for her extremely hard work organizing this event, Mike Ward, our chair, and Dan Stein, Dean for Science, for their support of this event.
Come think that almost all what I wanted to say has been said already. In this or another way, even my slides were used. So if I will still give my talk, but if somebody feels like leaving, it will be understood. So the title that I wanted to use today is The Amazing Ribosome, Its Enemies and Its Origin. I put it on top of flowers because what was most important, more than all what Tamar showed you with the press conference, was the amount of flowers I got in the first few days. I want to share it with you, and I want in this way also to thank you for inviting me. I couldn't come with real flowers because the United States immigration doesn't let. Once they almost arrested me for half a clementine, so this is what you can get. So DNA is the storage of the genetic information. It's actually just a string of instructions, and it can become, or it can be used for making proteins, which are the cell workers, after being transcribed to messenger RNA. The difference between DNA and messenger RNA is not very large chemically. RNA has a different backbone, and DNA has 4 nucleotides, and RNA has 1 that is a little bit different. But the difference in behavior is dramatic. DNA is a double helical molecule that has the information between the 2 helices, and it's hardly available for any type of chemistry, whereas messenger RNA, all the RNAs can live relatively happily as single molecules, and therefore they can expose the information that is stored here to be translated into proteins. The translation from messenger RNA to proteins is done by the ribosome. I put all the transcription here in one little yellow box, as if it's not important, but please don't misunderstand, or please give it enough respect. Roger Kornberg got for this the Nobel Prize about 7 years ago. So I love this slide or this picture, although it's childish, because I think for whoever doesn't know yet what ribosome is, it explains very nicely. It's a factory that needs some energy, actually not much, that can read the instructions that come as a string on messenger RNA. And people of my age around here can remember that there was time that we gave the computers instructions on paper tapes, more or less the same way. The instructions are being read inside and can come out afterwards to code for another protein. The beads here that are brought on carriers called tRNA are becoming a protein by being connected to each other, and the tRNA can go out empty. I apologize for those that do it every day or read about it every day in the interest of those that don't. So this at the end is a protein. At least this is a way to draw it. Actually in life it is folded, and only the folding, the 3-dimensional type folding, allows it to do what it needs to do. It even allows it not to fold or to have different folds. And I really don't want to talk about proteins. I want to talk about the ribosome. So messenger RNA comes to when it comes to the ribosome, or when it's available in the cell, it's a long line. Of course, it has much more complicated structure than my little cartoon here, with 4 different bases. Each of them is called a letter, and triplets of them can code for specific amino acids. So when something like this comes to a ribosome to be translated, first of all, the ribosome has to decide what are the triplets to be translated, because there is a big difference between, for instance, these triplets, if it starts from here, or if one starts from just one nucleotide, one base over. And this is what the ribosome knows to do. It doesn't know to decide which messenger to translate, but it knows to decide where to start and what are the triplets. Ribosome has two subunits. I'll come back to it in a second. The small one is the brain. The small one knows where are or how to assign triplets and also where to start, where is the initial one. So the initial one has a specific sequence, but it has to find it. And once the specific sequence is found, the first tRNA that brings the amino acid, which is cognate to the first, can be bound. Until now, I drew it just on white background, but actually this happens on the ribosome. So I already said the small subunit knows how to assign the triplets, how to scan for the first one, and allows the tRNA to bind. It makes sure that the binding is correct and that there are as little flaws as possible. So we call it the brain, and the worker, the hands, are the large subunit on which the peptide bond, the bond between the amino acids, will be made. So here is a tRNA, the carrier that is actually quite large, not as large as the ribosome, but quite large, that can find its position for decoding here, and deposits the amino acid for making a peptide bond. And the way it's done, I have a real quick cartoon for that. The small subunit from the side is rather slim in profile, and people called it a duck in the past, the electron microscopist, with a head and a body and a neck. You'll see from the side that maybe it can really remind you of the duck. But anyway, around the neck, the messenger is placed. And this is the first position where the first tRNA will bind, what I just showed you, the initial one. It's called the P position. There are three sites, A, P, and E. P is where peptide bond will be made above the peptidyl transferase center that is here, where the amino acid here. There is also an A site for the amino acylated tRNA that will come afterwards, and an exit site here. So once the initial complex is being made, there are factors, I don't show them now. The large subunit can come. There is very nice complementarity between the two surfaces, but it's not enough. Conformational changes are happening, and they are making intersubunit bridges. Now the fit should be perfect. The amino acid is now at the P site sitting on top of a tunnel for which the newly born protein will come out. And we think that this tunnel is the way the ribosome protects the newly born protein until it is maybe not large enough to fold, but larger than nothing. Once the initiation complex binds to the large subunit, ribosome can start functioning. Another tRNA comes, it will move here. Peptide bond will be made here real quick. The first one can come out and a new one can come in, and the translocation that follows goes in this direction. So next one will be here a yellow amino acid. Now we have a little movie that we made right in the beginning when we saw the first structures. The movie actually was not made by us, was made by art students from Jerusalem, but it's based on our coordinates in comparison to others. I want just to mention it was done in 2002 when the first structures came. So here is the messenger arriving to the small subunit. There is here a factor, IF3. I actually started with it. And now the large subunit can come with the first amino acid, also brought by a factor. Factors leave, the first tRNA comes, now the large subunit comes, makes bridges. And this is already the active ribosome. This ribosome can make proteins. More and more tRNAs come, and they stand in line, getting in according to the sequence. Protein is being made here. The ribosome helps. It has lots of flexibility, getting out, getting in, getting out, getting in, getting out. I want to tell you that this, what we see as hands, for those people that have some experience with flexibility of biological materials, these are 100 nucleotides and at least one protein. On one side there are 4 proteins. It all moves. And I'll come back to the speed in a minute, to the pace in a minute. I just want to show that the protein comes out from the other side and folds either alone or probably more like with the help of chaperones. The whole thing continues until there is a stop codon. Then recycle factors and release factors are replacing the tRNA, and the 2 subunits can dissociate. So there are many, many ribosomes in each cell. In normal mammalian cells, they can reach 1 million, and in very highly active ones, like the liver, they can reach 6 million ribosomes working at the same time in one cell, all the time, making proteins almost constantly. Even bacteria can reach 80,000 ribosomes working all the time. It means they are really doing a lot of work. And they are doing it very quickly. I promised you to talk about speed or about pace. 5 to 15 bonds per second. 5 in mammalian, 15 in bacteria. I needed a day being a student, a good student. So it's really incredible speed with hardly any mistake. There are mistakes, but the ribosome cannot even proofread them. But most of the mistakes either don't occur or the ribosome proofread. It's about 1 to 1 million. It's a very small number, but from point of view of cell life, it's still significant. So that's what the ribosome does. Even in the 70s, it was already shown that ribosomes are made of 2 subunits, and that many ribosomes work on the same messenger RNA. It means they scan it throughout, and that the proteins come out from the other side. This is an electron microscope from the 70s. I didn't take it. I found it in the literature without any bar and without too much explanation who did what. But even here you can see messenger goes between. Small subunit is bound to the messenger. Maybe here you can see, and protein comes out from the other side. This was all known when we started. What I didn't tell you yet, in the ribosome, ribosome is not just two gray bodies. They are made of many components. Small subunit in bacteria has molecular weight of 850. One very large, very long RNA chain of about 1,500 nucleotides and 20 different proteins. Large subunit has 2 long RNAs. One is 2,800, the other about 280, and up to 35 proteins with a molecular weight of 1.5 million. In mammalian, the ribosomes are larger. They can reach 4 million in molecular weight. The structure, though, the structures that are known now are still not of anything else but bacteria, but very complicated. Before I go to the structures, small subunit, I want to repeat, is responsible for initiation, for choosing the messenger RNA decoding and fidelity control, and the large one for peptide bond formation and for getting the newly born protein out. So actually this is the way what you saw in the movie. There are always 3 tRNAs bound. Here's small subunit, large subunit. PTC is where peptide bond is being made. This is, if you can see, the messenger RNA. A and P are decoding here, and peptide bond is being made there. And if you want to see it in more detail, although Tami already showed you, here it is, the large subunit, the small subunit rotating. You see it's a duck? I'll tell you again. Again. A duck? Well, I think it's a Snoopy, but nobody asked me. They gave the names before I went into this field. And the large subunit is called the crown. And maybe now it looks more or less like a crown. See, the king of Sweden doesn't have a crown. It's only the queen and the daughter, the princess. On the way, I will talk about the way afterwards, but on the way to solve the structure, we tried to find some starting models, and we thought about doing it from two-dimensional sheets by electron microscopy, three-dimensional reconstruction. It took us 4 months to get the first microcrystals. I'll come back to it. And 4.5 years to get these just monolayers. But once we found a way to do it, we could get them in 10 seconds. So these monolayers, when they were what is called in electron microscopy filtered, had the shape of the large subunit. We're talking here in the large subunit. And we could do reconstruction, which was not done before. And we saw something that we call the tunnel. So if you want to see, we saw something like this, and this is a tunnel. Until then, the tunnel was announced in the literature, or at least was suggested, first by Malkin and Rich, 20 years before we visualized it, based on biochemistry, and 3 years later based on more sophisticated biochemistry by Sabatini and Global. These two papers were very well filed in people's drawers or in garbage or I don't know where. It didn't get into textbooks, and I, being already 7 years in ribosome field, was not aware of it. But I had to convince myself that what I see here, you see it like from here or from top, that based on those that you saw a minute ago, I had to convince myself. I repeated this reconstruction 13 times, and then when I was sure, I went to the literature and I found out that there is a tunnel. So at least that I can see it. I didn't tell you yet, but when I started with ribosome crystallography, I was not well respected all over the world. Some people did, but most of them had questions or skepticism or worse about these studies. And coming out with, so I was Ada the village fool, or Ada the dreamer, or whatever you want to tell me now. I remember when I had the tunnel, I became Ada with the hole. And it took almost 9 years until it was rediscovered by cryo-electron microscopy. So now everybody knows there is a tunnel. I want to tell you, we could not convince the world that there is a tunnel, even when we saw it in the whole ribosome, not only like here in the large subunit. And for instance, in '88, just a year later, even Peter Moore, who is a well-known ribosomologist, doubted it. I don't really want to read it now, but there were lots of doubts. But there is a tunnel. You know now that there is a tunnel. Here is the large subunit. From the PTC to the end, this is the tunnel. We modeled into it polyalanine so we can see where it is, and we can show in this this color orange. And I highlighted here part of the tunnel to show that somewhere there are places that it's very wide and others where there are constrictions. And we think that the very wild parts of the tunnel can host partially folded proteins, and based on some work that was published during the last, I think, decade and a half, that show that for hydrophobic proteins, there might be some beginning of folding within the ribosome. So this crevice, we think, could be the position where such folding starts. I don't know what will happen to such folding when it reaches this constriction here. And this is still open. I'm not even sure that there is folding, but we have some, I'll show you in a minute some evidence for that. Where the constriction was cleverly chosen by antibiotics to block the tunnel, mainly macrolides, which is a very important antibiotic family that targets the ribosomes. So first of all, the crevice, we could bind a hydrophobic molecule into the crevice, and it's like a circular hydrophobic amino acids, and if one is ready to look together with me, so here is the modeled polyalanine, and that's the crevice. You see it's on the side. The protein can still go through the tunnel, at least until here. And something like this may happen, that instead of going straight, it can come here, fold a bit, and come back. So that's, I think, the only and still the most impressive possibility for ribosome to work as chaperoning, or to have some chaperoning properties. Let's look now at the constriction, where antibiotics bind. So because of the fundamental role played by the ribosome, a lot of antibiotics target it. Antibiotics, you know, this is the weapon one bacteria or one microorganism is developing against another microorganism. They don't do this for us, for using it in medicine, but in the middle of last century, it started. The use of it started. So antibiotics are like David and Goliath at least in our way of thinking. Bacterial ribosomes, those that are the target for antibiotics, are about 2.5 million Daltons, and antibiotics are most of them are about 800. They can reach 600 or 1,000. And the way they bind to the ribosome, or the way they stop the ribosome working, is more or less the way that the stone from David paralyzed Goliath. It hit it in an exposed position, which is important functionally. So this is the way the antibiotic works. It's not that just bind to the ribosome, but it has to bind to a functional site. And maybe I should say now, I should have said it earlier, all the functional sites of the ribosomes are RNA built, are solely RNA. PTC, it means where peptide bond is being made, also where the decoding is. RNA sites, means it's a ribozyme, and the antibiotics are mainly bound to RNA pieces. So here you can see 3 antibiotics that I used. I put, all what you see are crystallographic results. It means it's not just imagination. I put them into the structure of the large subunit where the RNA here is in bluish, and the proteins, all the proteins are in greenish, and the positions of the antibiotics, the magenta is at the active site, where the A site tRNA binds. The yellow is exactly where the peptide bond is made. I think you might have hit these names, not only the doctors, but also those that go just to pharmacy. Chloramphenicol, maybe less. It's not very useful. Clindamycin, maybe less because it's very, very potent. But erythromycin is a name that is more sold, and it sits at the entrance to the tunnel in the constriction here. Now, erythromycin is the mother or the grandmother of a whole family. So let's look at it and maybe I'll say some words about it. Here is erythromycin looking into the tunnel. The ribosome here is in all types of green, RNA in dark, and proteins in light. And the erythromycin here, sitting, you're looking into the tunnel from top, and it blocks 2/3 of the tunnel. It means it just doesn't let the amino acid, the newly born protein go through it. From the side, it looks like that. So that's a slice of the large subunit in a dash. T-site tRNA at the PTC, the newly born protein would have gone here if erythromycin does not stop it. So that's the way it functions. It, as I said, it's a mother of a whole family, or grandmother, which is called macrolides. And I really don't want to go a lot into chemistry, although it's a chemistry conference or chemistry lecture, but just to highlight that all macrolides are made of a ring. Erythromycin has 14 members, 2 sugars, 1 amino sugar and a normal sugar. Erythromycin is a good antibiotic, but it's not very stable in acid environments, and the stomach is an acid environment. So for treating problems in the stomach, companies try to make it more stable in acid environment, and they made 2 specific modifications in here and in here. Here and here. Here aliphatic, long aliphatic ring, and here just a methyl. That's not so important. The names of the results of the new compounds is also less important, but I will mention them. So let's look what happened just with these little changes. This is a view into the tunnel. You remember the arm that was jumping in, out is shown here. And now I'm showing here just the RNA now. I also didn't say that RNA occupies 2/3 of each ribosome, except for mitochondria, where part of the RNA is changed by proteins. But let's look into the tunnel entrance, and this is the way erythromycin binds. Now you see it atom by atom. Clarithromycin, which is just a little bit larger and more stable in acid, is even better occupying the void that is still available for proteins. And roxithromycin binds with two new anchors, and we dare to think that we see here structural basis for dosing. So I talked to several doctors that are treating infectious diseases, and they say that in many cases, the effect of 150 milligrams of roxithromycin twice daily for only 4 days is equivalent to what one gets with 500 milligrams of erythromycin 4 times a day for 7 days. I'm sure it's not all cases, but there are cases like this. And we dare to say that this is because erythromycin binds not as strong and as long as roxithromycin can bind. Now, over 40% of the antibiotics disturb protein biosynthesis, and most of them bind to the ribosome, and there are problems. There are problems that are connected to resistance and to selectivity. Almost, I think, all antibiotics by now induce resistance, slower or faster, and on this we can discuss. So all antibiotics, I said earlier, bind to the ribosome at functional sites. But ribosomes, functional sites, are highly conserved. So how do the antibiotics differentiate between pathogens and patients when these sites where they bind are highly conserved? So one way, the most popular way for antibiotics is very subtle differences. You want to see a subtle difference? So let's look at the tunnel here. These are the tunnel walls in gray. And structures of many antibiotics, many macrolides, catalytes and azolides, all the daughters of the macrolides are bound here. Everything you see here are crystallographically determined. All of them interact with one particular base or nucleotide on the tunnel wall, which is its number, running number in the Bible of the ribosomes, E. coli ribosome is 2058, we call it 2058, and it's adenine in all eubacteria. All pathogens are eubacteria. So this is the way it is. Adenine, or A, in eubacteria, that's the way it sits. And here is erythromycin. And all contacts here, or interactions here, are favorable. Very, very nice binding. Now, please pay attention to this position here. That's the way we are done, we are built, or everything above our here. Just instead of A, it's G. That's the only difference between bacteria and the patient. And this is now too short here, this distance, and therefore there is rejection. There is no binding. So that's the way erythromycin and its daughters and granddaughters bind to pathogens and not to human. Now, how do the antibiotics acquire resistance? Prominent mechanism is to modify the anchor. The subtle differences that they distinguish between eubacteria and patients are the target of changes. For instance, the selectivity that I just talked about is A in eubacteria and G in eukaryotes here in 2058. And that's erythromycin. We look at the same figure I showed you earlier from the other side. All what I had to do is change the word here, selectivity, by resistance. Now, the resistance here is either A to G mutation, pre-translational, or post-translational air modifications here. It means methylation of this nucleotide, and then there is resistance. So it's just taking advantage of what we took advantage in order to not to bind to patients. We're lucky that there are more structures than just ours. We are talking about eubacteria or infectious bacteria model, the Anococcus radiodurans, but between bacteria and human, there is some other branches, and one of them is archaea. Archaea is also bacteria, and in the particular case that I want to talk about is an archaea that is a bacteria that grows at the Dead Sea. So there are two bacteria that grow at the Dead Sea. You can see the Dead Sea from top here. From closer here, the Dead Sea is full of salt, mainly sodium chloride, some others too. Saturated, salt even comes out. And bacteria, the Heloarcula maris morti, for instance, grows on the salt. So in the winter, or what we call in Israel the winter, the height of water was higher than this piece of salt, and bacteria grew, but now it's summer and they're still there, and you want to be more convinced, look at that. Still bacteria grows here. So this bacteria is archaea, and ribosomes from this bacteria, the large subunit, were crystallized, and the structure was determined at Yale. Because it's archaea, it is a model for patients. So the Heloarcula, that I call now H, that was determined at Yale, is a patient model, and the Deinococcus that was determined by us is a pathogen model. Now, why do I tell you about this? First, because there are antibiotics bound to this one and to this one, we can compare. Second, we can see that even to archaea, not all antibiotics that bind to bacteria bind because they are like patients. So sometimes they bind under concentrations that are not acceptable medically, and sometimes they bind otherwise. And this is what I want to show you. The companies, when they discover that there are resistant strains to erythromycin and its daughters, decided to make new antibiotics that will bind to the resistant strains. When I heard about it, it was already in the works, but when I heard about it, I wasn't happy at all, because I thought if it binds to resistance resistant pathogen, it will also bind to the patient. But luckily, at least in the successful cases, I was wrong. So for instance, it's a successful case is here, azithromycin, that has in the main ring, instead of 14 members, it has 15. It binds to Haloarcula, and it binds to Deinococcus in green and in blue here. But when we superposed one on top of the other, I understood why I was wrong. In Haloarcula, it binds along the tunnel axis, whereas in Deinococcus, it binds across. So from this, we studied something very important for development of new antibiotics. Binding is not synonymous to inhibitory activity. It can bind, but it is not always an inhibitor or an antibiotic. It shows that there is binding, but not the binding mode, not the efficiency, and not the clinical usefulness. And we are now working on these aspects quite a lot. So let's now talk a little bit about the PTC, where peptide bond is being made. Just to remind you, here is the tRNA. The codes here makes the peptide bond here from the amino acid that is bound to the other side of the tRNA. So it looks like this. Peptide bond is being made there, and from there it goes to the tunnel. There are antibiotics that bind also and are efficient also at the active site, although the active site is almost fully conserved. How this happens? Let's look at one family as an example, fluoromethylene, that has a ring which is common to all its daughters with many, many types of hands. All types of chemistry, and if one looks at this chemistry, one cannot even imagine that all of them will bind the same way. Well, they do. How does this happen? First of all, have a look how nice it is. For instance, here, the ring binds the same way, but every side chain binds differently. You have here four examples together. Here, together with the A and P tRNAs, just to see here the ring, and everyone here is very different. What happens is that if we look only at the hand, for instance, the red, the ring will be here out of the picture. It comes into the binding site, and it would have banged into that nucleotide in black. But this nucleotide is very flexible, and it moves to here. When it arrives here, it attracts this one that moves here. So now the green one is the new position. There is now a gate. Antibiotics does not touch anything, just sits there, and the ribosome provides a gate that was not before, that the antibiotic cannot go out. These motions are carrying more motions in the second and third level. There, the level of conservation is lower, and therefore there is a difference between binding to bacteria and to human. So this is the new structure, and this is correct always. What we found is the cross-resistance to antibiotics also happens in the second and third layers, sometimes even 8 angstroms away from where the antibiotic binds, and always on the same side. So if we look at the antibiotic as a balloon here, only cross-resistance and changes are on this side. It shows that first, these are very important, and second, we could find that there is one specific nucleotide that attracts all these changes and transmits it to the ribosome. So that's the way there is binding and usefulness in the PTC, but also resistance there. So the ways that the people are using for combating antibiotic resistance are new compounds with additional anchors and drugs that are made of 2 compounds, and I want to show you 2 compounds, synergism between them. One of them came into the market, I think 6 or 7 years ago. It's made of A and B streptogramins. It's called synercid. And the clue is, here is the tunnel wall, one of them, one component, second component. Now it's very difficult for each component to go out, and there are also many more anchors. When synercid came out, everybody said there will not be resistance to it. Now there is, but it took really much longer time, and it's very few cases of resistance. So maybe there is a point in it. And it might be a little bit happier. You see here? That's the way I looked before, and this is after. But not much happier. Anyway, we are trying now to work on this point, and I just want to show you one very interesting observation we made recently. There is a couple made of one by one organism in Sri Lanka. And this is why they are called Lankacidin and Lankamycin. And they are on the same plasmid, and we decided there must be some synergism between them. But all the companies that had this just rejected this couple and said it's not strong, it's not good, it's whatever. We got it was forgotten. So recently we showed that not only they shouldn't be forgotten, but that there is synergism between them. And we found out, more than this, that erythromycin, that looks almost the same like lancomycin, almost the same, there is hardly any change, has completely different binding mode. It competes with lankacidin, whereas lancomycin interacts and is synergetic to lankacidin. It means just very, very small structural differences can turn competition into cooperativity, and I think this is the best story I can tell you now in terms of antibiotics, and we are working on that. So to summarize, all antibiotics bind to the ribosome at functional sites. There is diversity and there is similarity. The similarity in binding is very welcome by the doctors because it gives a broad spectrum. The diversity allows selectivity. So when there is resistance to one, it doesn't say that it will be to the other two. I want to come back to the PTC for a second. First, I want to tell you that we identified around it something that we think is the remnant of an ancient prebiotic molecular apparatus that is still sitting in the contemporary ribosome. In order to explain it, please focus on this part in the large subunit. There is here a substrate or substrate analog. So this part is actually representing 180 nucleotides that are made of two sub-regions that relate to each other by an imaginary rotation axis. It's only the backbone that relates and the orientation of the nucleotide. There is no nucleotide similarity in terms of sequence. It exists in all known structures. I have here 5. Maybe I'll tell you who did what so you know who are the main people. Deinococcus radiodurans is ours. Helloarcula, Yale, Stites, and Moore. E. coli 70S, E. coli the whole ribosome made by Kate in Berkeley, and 2 structures of the whole ribosome from Thermus thermophilus, one from Santa Cruz and the other from MRC. They all are superposed on each other. And I want to tell you there are 30 more structures with other antibiotics or substrate analogs. They're all superposed too, but I didn't want to make a too complicated structure. We looked at how a substrate analog binds to the PTC. The PTC is here. I don't want to say much, but I want to say that the region that connects the end of the substrate analog, which is totally conserved throughout evolution, CA amino acid. This region is this bond, that connects this region to the rest of the tRNA, which is double helical, is almost superposed on the rotation axis, this one here. So this gave us the idea or the suggestion that the RNA, the tRNA motion from A to P is made of two components, a shift of the helical part and a rotation of the 3' end, the CCA amino acid, like that. The whole thing moves sideways and this rotates, and this rotation is confined and guided by the ribosome components, like you can see here. The volume from A to P will be from here to here. Peptide bond will be made around behind this part, and this is the ribosome in gray or in silver. And you can see that nothing else can happen except for the rotation. When the rotation happens, the rotating moiety from A to P, from blue to green, is making contacts with the ribosome in red, and the contacts are shown by the blinking, the yellow blinking. And this type of rotation was verified by comprehensive mutagenesis in the group of Suzuki in Tokyo, because they showed that all the components that are interacting with the rotating moiety are conserved, depending if its basis or backbone is the level of conservation. This rotation allows also the ribosome to be polymerized. I'll come back to it in a minute, because for many years the idea was that people looked at making a peptide bond. The ribosome makes a peptide bond. That's not enough. The ribosome has to make the next peptide bond and the next one. It's not enough to make just one in order to make a protein. So together with Lou Massa, who is sitting here, we looked at what could happen during the rotation, or in other words, when the transition state of the reaction happens. And we found, or actually they found, by quantum mechanics, that it happens while the rotation happens. So this means that the A site reaches here, a new A site tRNA can come in. And then it can go out, and it can continue to act as a polymerase in this position. People didn't like this one either, so I was Ada with the dreamer and Ada with the hole. Then I had Ada with the rotation, until several structures came into the literature with substrates in the A site and the P site, and you can see how wonderful the derived position of the P site tRNA, which also has here sort of derived analog, amino acid analog, is in good agreement with whatever was seen in the literature. So now the rotation mechanism is more agreeable, and it, as I said earlier, it allows the ribosome to act as a polymerase, create peptide bonds, and elongate them. So this is now this region within the large subunit. It connects not only one side to another, but also the two moving hands. It's directly connected chemically, and also the decoding site through the tRNA and through a bridge. So we think that it may be transmitting messages from one side to the other. Because think about it. Here now there is a peptide bond being made. The tRNA wants to go out as an exit. How will this next one know to come in? How will the messenger know to move? Something has to transmit message. There is a peptide bond, go on. And we think that this happens by this region. Another thing I can tell you about the region is that it has very, very, very high conservation. 98% conservation over 930 species that were known, from bacteria to elephant. And this is compared to the rest of the ribosome, which is called also highly conserved, but it's only 36%. So this indicates that the symmetry is beyond environmental conditions, And this suggests that the protoribosome, there was a protoribosome, and it was a single dimeric RNA enzyme, and it's still embedded in the core of the contemporary ribosome. So we hypothesized that if this happened, there were pieces of RNA in the RNA well that got together and made a pocket. You can see it from top or from the side. And this pocket grew afterwards to become a ribosome. So it's not enough to hypothesize and even to publish. We are trying to make it. We plan to reconstruct, but actually we are doing it. So we either use pieces of the contemporary ribosome as the part that we think came together, or what we call primitive version, that any nucleotide is there. And we think that if we synthesize something like this, it should fall that way and then make the ribosome. And we're working on it. We have some new results, but not yet to show everybody. Later on, after the first ribosome was made, it went under all types of improvement on optimization to become the active site. And we are very happy that two groups not in touch with us at all, one in Montreal, Steinberg's group, last year published a paper based on looking how the ribosome is, how the components of the ribosomes are bound to each other in the modern ribosome, came out with the idea that the symmetrical region, which we call protoribosome, grew afterwards to become a whole ribosome. They did it on their own. They didn't care how the protoribosome was made or how the symmetrical region was made, but they showed this. And another group in Georgia Tech came out with the idea the ribosome can be peeled like an onion, but anyway, there is a center of it from where everything grew. So that's what we think about the origin. I just want to say a few words about the way we went, so you know why I was a dreamer. This is the ribosome, and in the 70s it was clear that it won't crystallize. It was published that it won't crystallize. It was based on crystallization attempts, mainly use ribosomes from E. coli, which are the beloved ribosomes by the ribosomologist until today, that failed in crystallization. The reasons were that they put ribosome is too large, too flexible, it's not homogeneous, it's whatever. I think that it's also because it deteriorates very quickly. And the deterioration makes it so that when people prepare ribosomes, they are already partially deteriorated. So the polar bears gave me a good idea. I read in a popular magazine that hibernating polar bears pack their ribosomes orderly just before their winter sleep, which shows that ribosomes can be orderly packed. It's against what was published, that they cannot. Why did they do it? So my idea was that while densely packed, ribosomes can maintain integrity and functional activity throughout the winter, which are for a few months. The speed that ribosomes deteriorate otherwise in the cell or in the test tube is a few days. So I thought that this is the way the ribosomes, the bears keep a pool of active ribosomes. So when they get up at the end of the winter and they have to do all types of things that they didn't do while they were sleeping, they will have enough workers, enough factories to make proteins, which they need to do, for instance, what they do here, or to run after food. So this was my lead, and I thought if we can find conditions under which ribosomes can keep integrity and find ribosomes that are more robust and less deteriorated while we are preparing them, we have a chance. So a technique how to keep ribosomes more active or regain activity was a procedure actually was published 20 years earlier at the Weizmann Institute by two people that were my teachers, Zamir Nelson and the student Miskin, and based on alcohols and warming up. and we used their procedure to prepare the ribosomes, also to keep. Which ribosomes to take? So we thought about robust bacteria, thermophiles, halophiles, as you could see before, radiophiles, and so on. So we have crystals. And actually, we got the first crystals just 4 months after we started, microcrystals, with very, very poor diffraction power, but powder, so diffraction of many of them that are used as powder. If you want to have some knowledge about size, from here to here is 0.1 millimeter, and even today with the new beam lines, I'm not sure that this will be large enough to be measured. But many of them together, I could put in a capillary. I waited 100 hours, beam stop failed, but I had here several rings, and I could measure them, and they were in a reasonable agreement with what was found for scattering of ribosomes in solution by several groups, including Klug and Zubai. And although we wanted to get this way, I was really happy that I could see this after such a short time. Then it took 20 years to get the structure, but it's a different story. After 4 years, we already had larger crystals, much larger crystals, and electron micrographs of sections of them negatively stained showed that there is the internal order that is very, very high, in my opinion, with each of these blobs, black blobs on the size of large cell unit. So the story is, that's the way we started. These little ones, sections of them looked okay. After 4 years, we actually could get several spots. See here, near the beam stop. Now, you're laughing. JMB published it with only one comment, which was minor. And I don't know how many of you here know the beamline scientist in Hamburg that was at that time at the EMBL beamline. His name is Hans Bartunik, a very cold man. To make him excited was very difficult. and I had lots of problems with him. But when he saw this, he took the film. At that time, people worked with him and ran to downtown to make a picture. You see, that's why we have this enlarged. So I thought that if Bartunik is excited, maybe there is something. There is a point in continuing. In 1990, we already had diffraction to the end, which is high resolution. But at that time, we found that after hardly any exposure, we already lost all the details that we needed. So in crystallography, for those that don't know, the further you are from the center, the higher the detail, and that's what we wanted, high detail. So we lost it all after a very short exposure. And I really don't want to go into the whole story, but we found a way to recover it in an experiment that was done at Stanford together with Hakon Hope, who is sitting here. So I wanted to pay attention to some. I really don't want to talk now about the procedure unless you want. But I want you to find out, to pay attention to me being very worried and also very tired because we worked for 2 days until we got a crystal in the beam, and to Professor Levy, who is a friend of mine in Stanford, who came and said, "You really think that you will get something from this saliva?" But anyway, we got. And I felt like Eureka, because it took like 2,500 crystals until we found that we have to do what we call cryobiocrystallography. And maybe, I won't tell you how we did it, but what was the rationale? I thought, and I think that it's normal to think so, when X-ray hits the crystal, it actually hits a biological tissue. It's like when we go to take Röntgen and we are trying to minimize the exposure and the operators are running behind walls. This is because X-rays are breaking bonds in biological tissues. The results of broken bonds are what we call free radicals, which are entities that are either looking for electrons or have too many electrons. Anyway, they're unhappy. So they're running to look for somebody else or some other component that can either contribute or take at least balance their electrochemistry. And meanwhile, they break more and more bonds. So I thought I cannot stop the first bond to be broken because X-ray will do this, but I can stop the running, the motion, or minimize it if we reduce the temperature dramatically, because then the free radicals will have less energy to run. So this was the idea. So as I told you, it took lots of crystals until we introduced it. So with the second small subunit structure, we needed half. And with the third one, the one that I talked most of the time, the Anococcus radiodurans, we just found freezing conditions immediately in the structure. So I felt like Eureka, but we didn't have many of those minutes. Anyway, in my opinion, it was a real Eureka, because if one looks at the PDB depositions between 72 and 2008, for instance, one sees that there is a big jump at around the end of the 80s. So what happened there? Cryobiocrystallography, method called MAD for phasing. I'm not sure that I'm the best to explain it here, because Wayne Hendrickson, that introduced it, is in the crowd, so I wasn't even there. And better detectors made this big jump in numbers. It's not only number of depositions, it means number of structures, it's also which type of crystals could be exposed. And crystals that were too small and too sensitive and to this and to that now can be measured. And that's, I think, a big, maybe not big, but impressive advance. And you see, even 22 years afterwards, I'm still happy about that. No, that's the cryo, you see, that's the way we did it. And we have crystals and also diffraction. So I want to spend a few seconds for thanks. First of all, to the Weizmann Institute for keeping up with me. I was not the easiest for them, not for myself maybe. To NIH, Max Planck Society, and the Kimmel Center at the Weizmann Institute. Helen is sitting here, for financing my what was called dream. To the members of my group that were dedicated and enthusiastic in good and in more like these bad times. For those that believed in us, first of all, Professor Wittmann from Berlin, that with him we started, and also helped in setting up the small institute we had afterwards in Hamburg that was mentioned here. He even gave the first ribosomes and a lot of support. Franceschi that replaced him, from Michael Seller, that was the Weizmann president, from our scientific advisory committee, John Kendrew, Chris Anfinsen, the late, both Nobel Prize winners, and Alex Rich. I want to cite exactly what Kendrew said. He said, when I started to solve the structure of myoglobin, everybody said it's not possible. So things like this shouldn't be stopped, and this is the best he could give me, which was really great. I want also to cite Chris Anfinsen that said, the real good ribosomes will be from my bacteria, which is the bacteria under the sea in hot springs, but nobody could grow it on Earth. And Alex Rich, who is still alive with us at MIT, that tried to crystallize ribosomes for 3 years, failed, and was still supportive. I also want to thank Helen Kimmel, who paved the way for us. Now, the provost already said that she's paving the way in NYU, so it's not only NYU. She's also doing it for us, and she has done it by setting up the Kimmel Center. And before I finish, I want to show you my group. I don't want to go one by one. Whoever is interested, here are the places where we measure. who supported us and so on and so forth. But pay attention, or maybe first to Yonath, who runs the group also when I'm here and when I'm there. And Tamar, who joined us 12 years ago for 10 weeks. She's now having her third baby, and she had a birthday when I took this picture. You see she has a cake in her hand? Maybe you don't see all the details, but I'll show you the cake in a minute. But anyway, it is like the large subunit, and it shows that in our group ribosomes are sweet. I also want to thank my family, and I'm doing it in favor of my family, of course, and also young women here that think, shall they stay in science or not because of family obligations. So my family supported me throughout, and Tamar showed you already in the movie. Although not always I was with them. My best prize, among all the prizes, is the Grandma of the Year. Tamar showed it to you, but what she didn't say is this prize was given to me when she was 10, now she is 14. It's still here, although there is no year. And when I asked her, why didn't you write a year? She said, because you have to reprove yourself every year. So it's demanding, not only loving family, also demanding, and it's already 4 years with me. See, here is the cake, sorry you don't hear. And Tamar already showed you what is now happening in the country. It's called in Hebrew Rosh Maleh Ribosomim, and in English the translation will be head full of ribosomes. And this is, I don't know if a punishment or a prize for the fact that I didn't go to the hairdresser before the ceremony, and the whole country was interested. What will you do? What will you do? So now curly hair is Rosh Maleh Ribosomim, and you see each of them is a ribosome. I don't know the person, Kishka, I never met. But you see here is a ribosome with an antibiotic in it. So thank you. Thanks to you.
Very much, Ada, for showing that dreams can become a reality. And I'm sure there are some questions that Ada would be glad to answer. Science and otherwise. Yeah, Suresh.
Great talk. I really enjoyed looking at the mechanism that we know about receive that my question is, looking at you did show in 3D as well as in your 2D structure of the RNA, the P and the A site. Now the E site you didn't show. Is it that E is symmetrical to A and you can't really tell, or they are distinct?
E is free. It goes out. It doesn't have an amino acid anymore. It is not involved in making peptide bonds. So it's not part of where things happen. It just goes out. Okay. So it is a tRNA, but it's not sitting at the symmetrical region. It's on its way out. And actually for the E site, there are several papers, and those that publish it, I'm not part of it, are still in fight with each other, who is more right or less, and I think that they're all right, except because E can be caught in different positions while it moves out. And even it's not clear whether it does or does not have triplet that it did decode at the time. It means that it's still bound to the triplet or not. And I think it's just because E is on its way out, and it's the structure were captured on different moments. But it's not part, it used to be P before it became an E. And then it's not part of the symmetrical region. It probably was not part of the protoribosome. We don't think the protoribosome had tRNA in it, just had a little, maybe 1 or 2 nucleotides and amino acid bound to it.
All right, thank you.
Any other questions?
Can you just talk about what happens when you go to Sweden to Stockholm at the prize? What actually What do they do?
You want to get ready to it? I already got emails like this. I want to become ready. What happens there? So I don't know what really happens there. It's for me, I felt like Alice in Wonderland. And people say that in all the pictures I look smiley and very easy. Because for me it was like a lot of happenings. Not all the winners were in a smiley mood. Some of them were very, very tense. In principle, what happens is that it's in 10th of December every year, because this is when Nobel died in its death. And Stockholm is quite dark, very cold, and even in the hours that should be some sun, it's usually full of cloud and either snow or rain. So there are only a few hours that have light, and the city is very happy with this happening. For them, it's a big thing. They have lots of TV shows. There are a lot of people standing in front of the hotel. It's the same hotel every year, asking for autographs. They stand there morning and evening, and the guards of the hotel already know who is more serious and who is less. And there are some that I was with a smile giving as many autographs as they wanted. One said he's already standing there for 31 years. Wow. every winter, so probably has nothing else to do. But more than this, the suites are very organized, extremely organized, and every minute is planned, and there are very few minutes for ourselves. The number of people that can come with you, and where you sit in the limousine, and when does the chauffeur opens it, and how you get in, it's all planned. And I was not didn't realize it in the beginning. So we had some discussions with the chauffeur. But in my last, you see, each category has to choose one person to talk for the category, 2 minutes in the big banquet. So somehow the two people that share the Nobel with me, one American and one American that is now in England, both speak English as mother language, decided that I should do this. I don't know why. Maybe they were told to decide this. I don't know. I have no idea. But I was stuck with it. And before the day that, first of all, one has to write these 2 minutes, and it has to be authorized. But before this, I got some advice. So the advice was, don't thank anybody, because it will take you more than 2 minutes to thank, especially in our case, 3 groups, and there will be always somebody that will become upset and offended. So don't thank anybody. So I said, can I just thank the chauffeur? As a joke. And this was I had to sit the whole evening, a whole dinner with the head of the Swedish Academy, because I think they decided to tame me a bit, and I was sitting there, and this was most of the discussion, what I shouldn't do in my 2 minutes. So when I said about the chauffeur, he said, oh, the chauffeur is a good idea. So indeed, I thanked the chauffeur. And because I said that he was nice, and I try to say that all the suites are nice this way, not only the big shots. And because of him, we could make everywhere. We could come to all the happenings on time, and in this dark city, beautiful dark city. So, and when I came out, this was midnight. 21 chauffeurs were standing in line. Neither of them was inside, and they all took their hats off and said, thank you. Now, there was an attaché to everybody, like a chaperone, and she told me that she had very difficult time with the Israeli media, the TV and the radio. They were calling her every day. They want an interview. She had to manipulate it, to kick them out. But after I gave the thank you, the Swedish television started to call. They didn't want me anymore. They only wanted the driver, the chauffeur. He got afterwards, he was invited by the head of the academy, the head of the Ribosome, and there was a big newspaper article, 105 years for Nobel, and the first time the chauffeurs are thanked. So if you wanted, you got a story.
Okay, let's thank the speaker for a marvelous talk.